So when we are in the biotech lab, we have a multitude of applicable math problems that we need to consider with our theoretical math tools that we've gotten before in algebra or geometry or calculus or statistics or wherever we get them from. And sometimes they're not said explicitly, but these are some of the things that we need to be familiar with as we go forward. Now, here's something that in our educational biotech lab we have been working with a lot and troubleshooting a lot. So let's read our problem out and see what our current tools are for solving it. Using column chromatography, green fluorescent protein, also called GFP, can be extracted from transformed E. coli. The lysate from this clogs the HIC columns and must be diluted by a factor of 1 to 10 in 0 molar TE, and then mixed in a 1-to-1 ratio of lysate to 4 molar binding buffer, also called BB. If the column is being loaded with a total of 500 microliters of the 1 to 10 sample, how much of the original lysate is needed to dilute properly? So if you haven't worked with HIC before, it is the kind that we've been using in our lab, is gravity assisted. It looks like a syringe tube. It's transparent here, and you have the little stopcock going on down here. You load your samples into the top of it, which is open, and you try not to disturb this bed down here. We call it the resin bed, and this is what our protein actually binds onto. It's a bunch of tiny beads that have a special coating on them for a protein to bind onto them when they're put in the right buffers. For us, it's the 4 molar binding buffer. I think of it, the higher the molarity, it's more stuff, more of our salt in the mixture, so 4 molar, more stuff, more sticky. And so because this is so large, our 0 molar sample of lysate will actually bind to the beads in our resin bed in our HIC column, and we can use lower molarities to wash it off. And in the end, we want some green fluorescent protein coming down here, and this is the stuff we collect. So we do this a lot in the lab, and it's what we consider very simple math, but it can be very complicated to get started. So let's see what we have available to us. So the first thing that comes to mind is the 1 to 10 sample. Or let's start all the way at the top. Let's see what this looks like and then break it down from the top. So what we're looking at in the end is we want 500 microliters of our sample lysate, and we want that ultimately to be in a 1-to-1 ratio, meaning 500 microliters of our 4 capital M molar, I'm going to call it BB like I have it right here. So in the end, this is what we're actually going to be putting onto our column, but this step right here is what we are concerned about at the moment. So let's bring that down. We're not here yet. We have our sample lysate. Now our sample lysate, according to our problem, needs to be in a 1 to 10 0 molar TE ratio. Now what does that mean? That means we need 1 part to 10 total parts. Now we know that the 10 total parts is going to be our actual fluorescent protein and our lysate. Our lysate is very, very messy, which is why we're putting it through this filtration system. And so we want all the cell debris and anything else we can get off of it from the E. coli, and we want that messy stuff in with 0 molar TE. So this total is actually a combination of our diluent and the thing we're diluting. So the thing we're diluting is this 1 part right here. We say, okay, so we can consider it as a 1 to 10. We can say it's that our GFP is going to be 1/10 of the total, or we can say, let's not put that over there like that. Let's say 1/10 or we can say that our GFP is going to make up 10% of our total. We're making a total of 500 microliters. Now, if you haven't seen this before, we are going to be using, because it is a dilution problem, with dilution problems we are allowed to use this handy dandy little trick of C1V1 is equal to C2V2. And this is concentration 1 times volume 1 is equal to concentration 2 volume 2. Again, it's standard, but we take it for granted. This is for dilutions, and if you have done titrations, you know that you get in trouble for using this because it is not a dilution. But we are making a dilution, we can use C1V1, C2V2. Now, the tricky part can be figuring out what these actually stand for. So when we're starting out, I like to set aside the relevant information. Now let's see, we have concentration 1, yes, awesome, that's going to be this right here, our 10th, right? So if you can go straight, if this made sense to you, you can stop me right here, and you can just move forward. 10% of 500 microliters, sometimes it's not that clever, but for us, we are going from 0 to 10, so we're going to call that 10x for our quote-unquote concentrated stock, and we want to go down to always a 1x. Now the x just means by how many factors it is either more or less concentrated. So our stock is always more concentrated than our working. And the working is what we want to get to so we can put it into our HIC column. Now, V1 is the thing we're trying to figure out. We want to know what this 1 is, or 1 in 10, or 1 part to 10 total parts. We'll do total parts. So this is the thing we don't know. So I am going to just call it V. Make it nice and simple. I know it's volume. Our V2, on the other hand, was laid out for us. It's this 500 microliters. So we know our unit. We know that this volume should be in microliters because that's what we've been given this whole time. We don't have any conversions that we need to be doing, which is something that we need to consider as we get more complicated. This could have been milliliters and you end up needing to convert it to microliters, but for now, this is what we have. So we have 3 of the points of data that we need. We can use quote-unquote simple algebra. It is simple, but if it has either been a while or you're in it now, I can understand how it can be overwhelming. We're only solving for 1 variable. So we have C1 is equal to 10. I'm going to just drop the x because that's confusing, times V is equal to C2 is 1x, and V2 is, whoops, I'm sorry, there we go, to 1, we'll just leave that as 1, times 500. Now this one I will put the units on so that I remember them in the future. Your answers should always have units when it comes to math, and in the real world it is to save our skins. So we remember what we're actually doing. So we go, okay, 10 times V is equal to 1 times 500. Now I like to do all the algebraic work and then do the calculations later because I don't like to either lose decimal places or what have you. So making this nice and simple, I'm going to wrap this up. I know 1 times 500 microliters is 500, but for sake of practice, I need to get V by itself. It's being multiplied. The opposite of multiply is divide. So I'm going to say V is equal to 500 microliters divided by 10, no units, is equal to 50 microliters of concentrated lysate. And this is correct. I need 50 microliters of concentrated lysate. And then what I would do from there, since this is a lab problem, from here we would take probably 1 ml, 1000 microliter epitube, or a 1.5 epitube, and it would be 50 microliters of our concentrated lysate. Let's go of GFP. So 50 microliters GFP, and then QS to 500 total microliters of sample. So actually it would be 450 microliters of TE. So in total, this comes out to 500 microliters of volume. 450 of it is TE. 50 microliters is GFP, and we know that it's a 1 to 10 dilution, and we can double check ourselves right here. So we can check it using a percentage. So let's just do our check right here. If you put the portion, right, so we're going to do a fraction to check it, because I know a 1 to 10 means 1 part in 10, which is about 10%, or it's exactly 10%, pardon. So I know that it should be 10%. So what is my portion? My portion is the 50 microliters of GFP, and in total I need how much? I need 500 microliters of sample lysate. And if I can do one of these, do one of those, 550, whatever you want to do. And I know it ends up being 0.10 times 100. We're just a mess right here. Times 100 is equal to 10%. So I know I did this right, and since this is a lab class, I'm going to figure out very quickly anyway, because I will not get any of that. So if no GFP comes off later, then you know you did this part all wrong. And unfortunately, that happens sometimes. Or it could be something else and you have to troubleshoot it. So I hope this was helpful, and you can go and purify your samples now.